Good afternoon. We are going to be continuing with lecture six today. You guys are my third section on this thing, and I was not able to finish for both of my other classes. So we'll do as much as we can, and whatever we don't finish, we will definitely wrap it up next week. Okay. Let's do a quick recap. We were learning about DNA replication, which is something that needs to happen before a cell divides, right? You need to copy all your genetic materials so that you can ensure they get passed on to the next generation, the next cell, right? Like if one cell splits into two, you want each of the daughter cells to have the same amount of genetic information. And so in order to do that, we follow the semi-conservative model of DNA replication, which was shown to be true through the Michelson and Stahl experiment. Right? So in the semi-conservative model, you separate the parental DNA and you use the each half of the DNA as a template to build the missing complementary strand. Okay? We started talking about this process in prokaryotes because that's how it was first studied, and then people did more research to figure out how it was done in eukaryotes. But let's finish talking about prokaryote first, and then I'll tell you the difference between what they do and what we do. In the process of DNA replication, there are several enzymes and proteins that are needed to complete the process. We talk about helicase, which is an enzyme that helps create the origin of replication. It creates the bubble. And then as it moves along the DNA, it will unzip the DNA further. But this unzipping action actually increases tension in the DNA double strand. And that's where you need to have another enzyme gyrase to help reduce that tension. The two strands of DNA are attracted to each other. The A's are attracted to the T's, and the C's are attracted to the G's because of the hydrogen bonding. But we don't want the two strands to come back together. We need access to these nucleotides. So that's where the single strand binding proteins are going to come in. They are basically going to keep the two strands apart and prevent them from coming back together. And there is actually a short form for this thing if you want to write it down, SSBP, single strand binding protein. In terms of the enzyme that actually builds the complementary strand, that would be the function of DNA polymerase 3. Okay. But there are two limitations when it comes to how DNA polymerase 3 works. Number one, it can only build on existing DNA strand. It's not going to be able to start the process on its own. Something has to come along and do a field-based pairing first, and then it can continue to work. Number two, it can only build in the 5' to 3' direction. And I'm going to add here because my other classes were very confused about this thing. Can only build in the 5' to 3' direction on the new strand. On the new strand. So not the black one, when we look at the numbers 5 to 3, not talking about the original, we're talking about the new one that you're building. That can only go in the direction of 5' to 3'. Okay? Now, I hopefully you guys can participate a little bit today because it based on the last two classes, it's very confusing for everybody, the thing I'm about to teach you. So I just need you guys to, tell me if you actually get it or not. Because if you don't get this next part, the rest of the lesson is not going to make sense. All right, so let me try my best for the third time. So DNA polymerase 3 is needed to build the nucleotides, right? So you put the T and the A and the, things like that. But like I said, it's not able to start the process on its own. Nobody knows why, but that's just the way it happens to be. Okay. So as an analogy, it's like I told you, it's like my kids, okay? One of them is learning multiplication at school right now. Okay, so for example, she come home and then with a worksheet, you don't have to copy this down. I'm just trying to give you an analogy to help you understand what's going on here. Okay. So let's say she has a worksheet, okay? So she talks it out and then she just sit there and not doing anything. So in order for me to help her, I would sit down and I would do a few questions with her. I'd be like, oh, 2 times 3 is 6, right? 6 times 7 is 42, and then this is 10. So I do a few questions with her, and then she will be able to do the rest on her own. Okay? But the thing is, the key thing here is she's not going to do the homework on her own. Someone needs to do a few questions with her first. And that's what DNA polymerase 3 is like. DNA polymerase 3 is like my daughter, not going to do the building of the DNA on its own. Something needs to come and do a few with it first. And that something is another enzyme. Okay, so this enzyme, you can draw whatever shape you like. I'm just going to draw a little diamond here. This enzyme is called primase. Okay, so the function of primase is to build build a short RNA primer such that DNA polymerase 3 can build upon it. Okay. Sorry, it's a little bit crowded. Let me see if I can move some of these things out of the way. I, of course, will post this whole thing after class as well, but I do want to continue writing from here. Okay, so there we go. So what does that mean exactly? Well, the primase is like me. Right? I'm going to come over. I'm going to do a few examples. All right. So T, what does T pair with? T pairs with A, right? And so that's easy. And then listen carefully. A pairs with T, right? We know A and T goes together. However, the primase here is actually building an RNA primer. Again, nobody really knows why. That's just how the body works, right? So instead of pairing A with the T, it is going to pair up with a uracil. Okay? And then C is going to pair with a G, and G is going to pair with a C. So this thing right here, this little short bit of RNA primer is necessary because of the limitation of polymerase 3. It's not able to build from scratch. It can only build on existing things. Is that clear? Give me some sign that you understand, okay? Or like if you don't understand, ask a question
Can you
before I.
repeat it again?
Sure. Do you
I
have a specific question about which aspect you want me
Yeah,
to?
you CGUA, you just explain it. Why
Oh,
A instead of T,
oh,
U instead
yeah,
of T?
right. So the primase, which is the square thing here, it's the enzyme that will build a little bit of nucleotide so that the DNA polymerase 3 can continue. But it doesn't actually build DNA. Nobody really knows why, but it just does build a little bit of RNA instead. So one of the differences between DNA and RNA nucleotides is that instead of having thymine, you have uracil, right? So because it's
Yes.
building RNA, that's why A no longer pairs with T, it pairs with uracil instead.
Okay. Yeah, got it.
Okay,
Thank you.
yeah, thank you. Everybody good so far? All right. Now I have another question for you. I'm gonna run a poll. Please participate if you can. If you don't see the poll, you can type me the answer. I just need to know what you guys are thinking so I know if you understand it. Now, the black strand here is the original DNA, right? It's the one we're trying to make a copy of. And of course, you should know by now that DNA have direction associated with it. So this black strand at the top is 5 prime to 3 prime. So I want to ask you, the primer, which also would have direction, what number should I put over here? Should I put A, 5 prime, or B, 3 prime? Please choose the answer that you think is correct. What do I put over here on the right side of the primer? Should I put a 5? Should I put a 3? Okay, anyone else? Has been more than a minute. Okay, it's about half half. Half the class is 5. The other half says it's 3. The correct answer is 5. All right, because it's anti-parallel. If you have two strands that are stuck together, you will never have the same number. If one of them is 3, the other must be 5, right? Because again, it's anti-parallel, right? They have to be running in opposite direction. So that's a 5 here, and that must be a 3 here. Okay? Now, next question. So back to the analogy, right? After I do a few questions, then my daughter is able to finish the rest of the sheet herself. And same thing here. After the primase put down a few primer, DNA polymerase 3 will be able to do the rest. But here's another question for you, right? In the second limitation we wrote down here, the polymerase 3 can only build in a 5 prime to 3 prime direction on the new strand. So do you think polymerase 3 will start putting letters this way, or will it start putting letter the other way? Okay, I'm gonna launch the poll for you. Is it this way? I would say A, or the other way, B. Let me relaunch the poll.
Sorry, can you repeat the question again? My Wi-Fi cut out.
Oh, no problem. So when DNA polymerase 3 continues to build the rest of the strand, my question is, does it build towards the direction of A, or does it build towards the direction of B? Okay, anyone else? The poll is anonymous in case you don't know, so do choose something if you are able to. Okay, again, we have a half-half situation here. The correct answer is A. Okay, it's gonna go towards the left side. Why? Because it says so here, right? The enzyme can only build in the 5 prime to 3 prime direction on the new strand. We are not looking at the numbers on the black one. We are looking at the new one. Right? So if you look at the new one, 5 is over here, 3 is here. So 5 to 3 direction is this way.
Sir, I understood that part, but I have a question for another part.
Sure,
So
yeah.
let's say that the primase is where it is now. Does that mean that like is there only one? Because let's say the 5 goes to the 3, but then
Yep.
what happens behind the 5? Like is there going to be another one making their other ones, or is that just
Yeah,
stop
yeah, yeah.
at the 5?
Good question. Good question. Hold your thought. It will come. Okay. Yeah. We're getting there. Okay. Very good observation. So it's gonna go from 5 prime to 3 prime, right? So the DNA polymerase 3 is gonna build more. Okay. C is gonna go with G. And again, please use color so that this is gonna make sense to you. Okay. Now we are putting T's now because it's DNA and not RNA anymore, right? So this is just going to keep on going and going and going. All right. Now I'm going to draw the enzyme here for DNA polymerase 3. Now, let's take a look at what's happening at the bottom. So I'm going to put another primer here. I'm not going to draw the enzyme. It's like already very busy, but I'm going to put a primer. I'm going to go like C, G, C, A, like that. In reality, the primer is much longer, but obviously we have a smaller diagram, so I'm just going to put a few here. Help me out. What number should I put here? Is it a 5 or is it a 3? Okay, I have a couple of 5s. Yep, it is definitely 5. Okay, 5 prime, because if you go all the way to the other side, you can see the bottom black strand is 3. So it's going to be 3, 3, 3, 3, 3 all the way. And you have to have the opposite number, right? And if this is 5 down here, this must be a 3. So once again, because of how the enzyme works, there's no why, it's just the way it is. So 5 prime to 3 prime direction. That means we're going to start building towards the right this time, right? T, C, and G. Okay. So that brings us to Michael's question. What about the rest of this stuff, right? Like, how are we going to copy this? Ideally, if the enzyme can move both ways, then, problem solved. But it doesn't, right? The DNA polymerase 3, once again, can only move in one direction, the 5 to 3 direction. It's back to my analogy, okay? Hopefully it's going to help you. Let's say, the worksheet actually was rolled up a little bit, okay? And you can actually unroll the worksheet, and then you discover, oh, there are actually more questions up here, okay? But suppose, let's just say my kid can only do question from top to bottom. Okay? So what's going to happen to all these questions? How am I going to make her do it? Well, I'm going to have to sit down with her and do a few with her again. And then she will be able to, go top to bottom and finish the sheet, right? So that happens every time we review more questions that are beforehand, right? And in the same sense, as helicase unzips the DNA as it reveals more nucleotides, it's not going to be a problem for the top strand because it would just keep on going into it, right? It's already going the 5 prime to 3 prime direction. But it's going to be a problem for the bottom strand. And to solve that problem, you basically have to put more nucleotides, more primers, right? So C, G, A, you're going to have an other RNA primer, and then the polymerase 3 will be able to fill the gap here. And then again, when you review more nucleotides, you're going to need to have another primer, okay? And then the polymerase 3 is going to go build more. And then more primer, more primer. Okay? So in a sense, the top strand, it's not a problem. It's just going to keep going, right? As the helicase unzip more, it's just going to keep on building the complementary strand. But for the bottom strand, you got to do it a little bit at a time, So some people ask, in my other class, why not wait until you unzip the entire DNA and then you just need one primer? Well, the whole DNA is really big, right? So you don't want to waste time, I guess, right? As it unzipped, you want to take that time to build at the same time. So you're doing two things at a time to speed up the overall process, basically. Any questions so far? All right. So let's zoom out a little bit and we will draw like a summary picture that hopefully will make it even more clear for you. A summary. I don't know what to call it. Summary, yeah, summary of DNA replication. Okay, so brand new diagram, doesn't have to be super big. Okay, so there we go. In the previous picture, we were only focusing on the one side of the bubble, right? It's called the replication fork. But now let's take a look at the entire bubble. I'm gonna put 5 prime to 3 prime here again, just like before. There is no reason why 5 to 3 has to be on the top. It could have been 3 to 5. Okay, but I'm just keeping it consistent so you don't get confused. 3 to 5 over here. All right. And once again, that's the parent DNA, the original parent DNA with the ACTGs that's not shown. Of course, the numbers are opposite to each other because of anti-parallel property of DNA. And you're gonna have your hydrogen bonds here and whatnot. Okay. In order to start copying the DNA, you need to put a primer. That's what this pink thing is. And it's made of RNA. Now, take a look here. If this is 3, then this must be 5. And then this must be 3, right? So that's the 5 to 3 direction, which means polymerase 3 is going to build like this. This strand here is being built continuously. And there's no breaks in it, no gaps. So it's called a leading strand. Leading strand. It's built continuously towards the replication fork. Okay? It's going to keep on going into it. Leading strand. At the bottom, we saw earlier, we need to, of course, put a primer as well, but this would be 5 to 3. And this time the polymerase 3 will build continuously to the right. So on the other side of the fork, this is the leading strand. Okay. It's going into the fork, towards the fork. The replication fork, that is. So what do we do with all these stuff that are being revealed as helicase unzipped more? Well, you're gonna need more primer, right? And more primer. And more primer. And then you'll be able to build a little bit at a time like this. So this strand that's being built a little bit at a time, that's being built in fragments, it's called a lagging strand. Built in fragments. Away from the replication fork. Okay. That's called towards the fork, that's called away from the fork. Right. Similarly, on the top side of the other half of the bubble, we have a similar situation. You're going to be building it in fragments. Fragment, 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 like that. And this would be the lagging strand. Okay. Questions?
So the leading strand is just the one that follows the current, and the lagging strand is the one that's behind it.
Sorry, one more time. I didn't quite catch your.
So like the leading strand is where like the strand goes towards the 5, and then the lagging strand is the one that goes from the 3.
Um, so when you say towards the 5 or towards the 3, it's best you use the new strand as the reference. Um, so actually the leading strand is going towards the 3, if anything. But actually they're all going towards the 3. Okay? What distinguishes them is whether it's built in one go or whether you have to do it a little bit at a time. That would be a better way to discern the two. Okay. Now these fragments have a name. These fragments, they are called Okazaki fragment, named after the scientists that discovered it. Japanese scientists here, Okazaki fragments. Okay. All right. We still have a little bit to talk about, and then we'll be done with this note. I'm gonna take this part here, and then I'm gonna zoom in for you. Okay, zoom in for you. Okay. So we have the 3' and the 5' here, and I'm just gonna make up some letter. Okay. Give you a moment to copy that down first. You see there's a primer here, right? The red thing. So I'm gonna put a primer here as well. Five to 3. And polymerase 3 is gonna come, and then it's gonna continuing on from the primer. Nothing's new so far. And then it's just gonna keep on going, right? Again, please use color for this note, otherwise it would not make any sense to you. So there is an other primer here. As helicase reveal more nucleotides, you're gonna need another primer, and then DNA polymerase 3 is gonna continue, right? Okay. So copy whatever's on the screen first, and then stop copying and listen to me explain first, right? Because I'm gonna be writing things down and I'm gonna be moving things around, erasing it afterwards, and then I'm gonna summarize it for you, okay? So this next part is better if you just watch and don't copy, okay? So this guy is basically DNA polymerase 3. Okay, don't write it down, okay? I'm gonna erase this later. Because you don't need it, okay? And then there's another guy. This is DNA polymerase 1, which I haven't told you about yet. So this is a situation. The primase, which is the enzyme that lays down the primer. This is a TAGT. That's the primer, right? It's there. And then the polymerase 3 is gonna hop on, and then it's gonna move along the DNA and build a strand, right? Again, that's old news. Nothing's new. CTTAGCT. But what happens is when the polymerase 3 gets to the primer in front of it, then it will fall off. And then it will signal polymerase 1 to take over. And the function of polymerase 1 is to remove the RNA primer and replace it with actual DNA. Okay, so one more time. As the DNA polymerase 3 approaches the primer, it falls off, and then polymerase 1 is going to take over. Right? Polymerase 1 is going to take over, and it will remove the primer and replace it with actual DNA. All right? So let me write that down for you. I gotta get my color sorted out. As what does U stand for? U is a uracil. Okay, it's only found in RNA. Okay, uracil. So as DNA polymerase 3 approaches the RNA primer, DNA polymerase 3 is replaced by DNA polymerase 1. And then polymerase 1's job is to remove the RNA primer and replace it with DNA nucleotide. And just as an aside, both DNA polymerase 3 and 1 have spell checking. That's actually what it's called. And autocorrect function. Okay, so when it makes a mistake, it's able to correct the mistake. It's like typing, right? You type and then you realize you made a mistake, you press backspace, and then you just fix the error. The polymerase 3 and 1 function pretty much in the same manner. Okay. So in the end, there are no RNA in the copies, of course, because we are copying DNA. And then to replace the primer, we need to have the polymerase 1. All right. Yes, go ahead.
What would be the rules for naming the nucleotides of the RNA primer?
What would be the rules for naming it? What do you mean by naming them?
By naming, I mean the full
Like the
form,
pairing?
the pairing.
Yeah, so pairing is here, I'll write down DNA slash RNA pairing. It's basically the same thing. T, okay, I'm just gonna write ACTG. And then for RNA, it will be UGAC. It's just the same with the exception that thymine, T is going to be replaced by U. That's the only difference. Is that your question or?
Yes, and it seems like for one of the uh, nucleotides in the example, A
Oh
is
yeah, I made a mistake, right? Here? Is it the? Oh yeah. I totally made a mistake. I'm so sorry, guys. This is thank you for pointing out. This is U and this is A. Yeah. And over here as well. That is U and that is A. Thank you. Didn't notice that at all. Okay, same thing here. There's another T here. It shouldn't be a T, it should be a U. Okay, and that's why we have to replace the primer with actual nucleotides because we cannot have uracil in the final DNA product. Is there anything else? Okay, so there's just one last enzyme that we're going to have to talk about. Once you replace all the primer, we need to bring in one more enzyme. Uh, lastly, I guess we can say lastly. Lastly, DNA ligase connects the Okazaki fragments together. By creating a covalent bond. Okay, and the process is complete. So I know it's a complicated process. I'm not going to ask you to explain it on a quiz or anything like that, but you do have to know what enzyme does what in order to answer some of the questions. Okay. So one more thing before we go back to the PowerPoint. Um, so I told you the prokaryotic DNA is actually circular, right? So it's double strand, of course, but I'm just gonna draw a single strand here to avoid confusion. And I just want to help you visualize this a little bit so that you understand. Okay, so there is the primer. I'm not gonna put the 5 and the 3. Just know that it's gonna go this way, right? And there's gonna be like another primer. There's gonna be multiple primers, right? And then the polymerase 3 is gonna build until it gets to the primer ahead and then falls off. Polymerase 1 is gonna take over. It's gonna replace the primer, so on and so forth. And what's good about circular DNA is that eventually it's going to loop back on itself. You're going to end up where you started, and you're going to replace the original primer. Okay? So keep this in mind as we move on to talk about eukaryotic DNA, because our DNA are not circular. Okay, ours are linear, and that actually creates a bit of a problem when we try to copy the DNA, as you will see. Okay, so again, the DNA is able to loop back on itself and replace the original primer. Yes, Amar, go ahead.
what would be the RNA primer replaced by when the DNA polymerase one attaches itself to it in the above example?
Yeah, so over here, right? The light blue here, it's basically going to take out the U and then it's going to put in the T, right? Because again, the equivalent of uracil in DNA is the thymine, right? So it's going to replace it with T. And then AGA, there's nothing wrong with it, but it seems that the enzyme actually will take them out and then put them in again. Okay? So the main purpose is just replace the U with the T. But all the other nucleotides, the A, C, and the G, they actually get replaced as well by DNA polymerase one, even though there's no need to do it, but it's still being done. Does that answer your question?
Yes, thank you.
Yeah. So Michael, I did not show the ligase in the diagram because it's already confusing enough. Okay, there's like 10 million things in the picture already, but basically it's gonna be a glue. You can think of it as glue that cause the fragments to go together. All right. Okay, so let's go back to the PowerPoint slides here. Okay, this is like the professional drawing. Does anybody want to guess what number two is here? What is this thing? What enzyme might that be?
The one with
Absolutely.
that opens the zipper.
Yeah,
I
yeah.
think it's helicase.
Yes, that is absolutely correct. Okay, so that's the helicase. Very good. It unzips the DNA. What about this one right here?
That's the gyrase, or
That
however
is
you
the
pronounce it.
very good. It relieves the tension, right? Okay, and number three, these little balls. Let me just look at your notes. Is all
The single strand binding
Strand.
proteins.
exactly right. Okay, so hopefully it makes sense, right? You just try to match these things up, and then here, you were looking for the ligase, and that's where it would have show up. Now there is a small mistake here. Usually people, I mean, it doesn't really matter, but like I just want to point it out so that in case you saw it, you will know. Okay, this is 3'. Don't draw this. Right? I'm just gonna follow the purple thing all the way, 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 like that. And then they did a little twist here just to make it fancy, but I think it's the twist that caused them to make a mistake. If this is the 3', then this wouldn't be 3, it would have been 5. Okay, and then this is 3. Okay, it's just a small thing. It's not a big deal. Uh, these fragments, right? Okazaki fragments. So what should we call this top one? Is that leading or lagging, huh? Leading or lagging?
Leading.
That is absolutely the leading because it's being built continuously, and then this one is lagging, right? But please remember on the other side of the bubble, the position of the leading and lagging switched, right? The top one is now the lagging, and then the bottom one is now the leading, right? So just be mindful of these things. Okay. So that all that stuff I taught you happens in prokaryotes, in bacteria, and in eukaryotes, which is us, right? And other things like plants and fungus and stuff like that. We do a very similar process, and then there are some key differences between what happens in eukaryote and prokaryotes. I'm going to highlight 3 differences for you. Number 1, eukaryotes have linear chromosomes, as I told you. So ours is not like a loop. Ours is more like, a straight line. And it's very long. Right? For example, in the E. coli, total genome, all of this DNA, it's only about 40 million base pairs, 40 million ACTGs. For humans, in a single cell, we have over 3 billions ACTGs. So in order to copy all of those things, it makes sense to have multiple origin of replication. Okay, so we don't just open up one bubble. We're going to open up multiple bubbles, right? As many as 100,000 of them. And then we'll let the bubble expand, and then that just speeds up the process. It's kind of like this, okay? Like if I asked you to copy like a children's book, right? No problem. You can do it on your own, right? You don't need help. But if I say, let's copy a textbook, right? Then the whole class is gonna have to get involved, and we are not gonna like all copy from the beginning, okay? Like maybe you're gonna start copying from chapter 2, you're gonna start copying from chapter 5, or like we have multiple people starting at multiple points, and then we just put the work together, right? That way you can simultaneously do more and finish quicker. That's what this is about, right? By having multiple replication bubble, you can copy more DNA at the same time and finish quicker. That's all it means. Okay, so that's the first difference. Prokaryotes have only one origin of replication, and it's fine because their DNA is not a lot. But for us, we have multiple origin of replication. Second difference between prokaryotes and eukaryotic DNA replication is that most of the enzymes that we've learned, the helicase, the gyrase, the polymerase, they have a eukaryote counterpart. And you're going to basically find the same enzyme in eukaryotes. But sometimes you have multiple versions of that same enzyme in eukaryotes. For example, in humans, it's not just polymerase 3 and 1. There are actually 5 DNA polymerases in total. Okay, and that makes sense because we are more complicated and there are more DNA to deal with. So it's not surprising you would have multiple versions of the same enzyme doing a job that's similar. Okay. And finally, the third difference, and it's something that we're going to spend some time talking about it, is that every time eukaryotes copy their DNA, and that includes us, okay? Every time we copy our DNA, we will lose a little bit of DNA. So if this is our DNA is linear, right? So let's say your DNA is this long, your chromosome is this long. If you copy it, then it gets shorter. If you copy it again, it gets shorter and shorter. So every time you copy, which is every time your cell divides, the DNA gets shorter and shorter and shorter. So of course you might wonder why. Will it die at some point? Yeah, we're gonna get to that. That is a very good question. Okay, but let's understand why it gets shorter every time first. Okay, so here we go. Another note. This is a short note. Why does linear DNA get shorter every time it's copied? Every time it gets copied. Okay. So I'm gonna draw linear DNA, and they are already separated. Okay. Like obviously, when it like in real life, you're not gonna separate the whole strand at the same time, right? You're gonna do the bubble thing, right? Okay. But for simplicity, I'm just gonna separate the whole thing to help you understand. I'm gonna put the 5 prime here and 3 prime here, 3 prime and 5 prime. Okay. So I'm just gonna start in the middle. I'm gonna put a primer here. I'm gonna put a primer here. Please think about this on your own. Which way are we gonna copy? Is it to the left or to the right? Let's do another poll. Let's see if it gets better. Let's see if I convince the other half of the class what the right answer is, okay? Let me go here. A or B this time. Okay, don't look back. Just think about what we've learned. And which way are we gonna go? To the left or to the right? Okay, I'm missing 6 answers. Choose something, please. Okay. Well, it's not the result I was hoping for, but that's okay. Let's try to talk about this one more time. We have the primer here, right? The primer is going to be anti-parallel to the black line being the original DNA. So if I have to put numbers on the primer, the numbers will have to be opposite to the black numbers. So if it's 5 on the left, it must be 3 on the left here. If it's 3 on the right for the black, it must be 5 here on the pink. And based on the limitation I told you about, DNA polymerase 3 is going to go in 5 to 3 direction, which means the correct answer is to the left. Okay, A. So what happens to these things? Well, you're gonna need more primer and more primer, right? So just draw it exactly the way I've drawn it, like that. Nothing's new so far. Just draw it out. And at the bottom, it will go to the right. Hopefully that makes sense. All right, this is 5, this is 3. The numbers have to be opposite to the black strand. And you can only go from 5 to 3 on the new one that you are building. All right? So you need more primer and more primer. So in the end, you would have copy each of the black strand. You basically you use the black strand as a template to create the missing complementary strand, right? Okay, so nothing's new so far. So what's the problem? Well, let's zoom in a little bit. This primer, which is made of RNA, is going to be replaced by DNA because polymerase 3 is going to come and then it's going to fall off and number 1's going to get on and then it's going to replace the primer, right? Same thing, this is going to get replaced. Number 3 falls off, number 1 gets on, replaces the primer. But we have a problem when we go to the end here. This little bit is not going to be replaced by DNA because there's no polymerase 3 coming from upstream. That's the proper term, okay? From above, right? From before. There's no polymerase 3 coming from upstream, and then there's no signal to tell number 1 to get on. And as a result, this little bit of RNA primer is not going to be replaced. The RNA primer at the end will not be replaced because no DNase polymerase 3 or 1 is coming. Okay. This part of the DNA does not get copied and will be lost. Do you understand why we will be losing that? It's not a problem for circular DNA because you just loop back on itself. But it's a problem for linear DNA because nothing's looping back. So you just end up losing the end. Right? Now you might think like, oh, the top one is okay at least, right? But it's not. Because if we go to the other side, same thing, right? Lost. will be lost. Right, so that's what I meant by every time you copy your DNA, it will get shorter and shorter and shorter. Now that, of course, is a concern, right? It's a very big problem. Imagine reading your textbook, but every time you read your textbook, you got to rip off, a couple pages from the front and from the back. Right? It might not be a big deal at the beginning, you lose your cover, that's fine. I can still read the rest of the textbook. You lose the table of content, the glossary, that probably is still going to be okay. But eventually you're going to rip into chapter 1, right? And maybe chapter 20 or something like that. And you're going to start losing important information, right? So same thing here, the DNA, which contains instruction to make proteins, if we start deleting those instructions, then you're not going to be able to make the proteins that you need for the cell to function properly, right? So we have to have a way to deal with this shortening. And there's no way to stop the shortening, but there is a way to kind of what's the word I'm looking for? Is that to cope with it, okay? To try to have it happen, but not cause any kind of problem. Yes, this is only a problem for eukaryotes because we have linear DNA as opposed to circular, right? Okay, so that's 1 hour already, if you can believe it. So let's take a break, and when we return, I will tell you all about what is being done to remediate this situation. Okay, see you in 5. Right, and we are back. So what kind of mechanisms are there in place to prevent the negative effects of this erosion that happens every time we try to copy our DNA? Well, it turns out located at the ends of each chromosomes, and we have 46 chromosomes in total. Located at the end of each chromosomes, we have these non-coding DNA sequences called telomeres. Okay? Telomeres. They are just repeats of 6 nucleotides. Okay, TTA, GGG, TTA, GGG, TTA, GGG, repeated many, many, many times. And they are located at the ends of each chromosome. So, there is actually a technique called FISH that can be used to highlight whatever part of the chromosome that you want. And in this particular picture, the technique was used to highlight the telomeres. And you can see that the glowing ends, that's where they are located. So think of the telomeres as the plastic thing that is at the end of your shoelaces. Right? So the plastic thing is there to protect the shoelace so it's not gonna fray and get damaged and separate, right? So in the same way, that's what the telomeres are for. So imagine every time you put on your shoes and tie your shoelaces, you have to cut off a little bit of the plastic thing. So you chop it off. Next time you wear the shoes, you got to chop it off again, and you got to chop it off. Every time you wear it, you got to chop it off. And eventually, you're going to run out of plastics, right? And so if you wear it again and you start cutting, you will cut into the actual shoelace, right? So that's basically what happens. Every time we copy the DNA, and that's something that must happen every time you divide a cell, and the DNA gets shorter. So you cut off a little bit of the telomeres, but that's okay. Telomeres don't code for anything. Okay, they are just there to delay the erosion of important DNA. Okay, so it just gets cut off, and eventually, though, it's gonna get to a point where the telomeres will be too short. Okay, so if the telomeres are too short, that's same as saying the plastic thing is too short, then the cell is not going to be allowed to divide again. Because if you go through another round of division, it means you will have to copy the DNA one more time. And that means you're going to start eating into actual coding, important DNA. So in that case, the cell is going to go into growth arrest. Right? So when telomeres are critically short, the cell is not allowed to divide further, to divide again. Right? It enters growth arrest. Okay? So as we age, more and more cells will not be allowed to divide anymore, and that's part of the reasons that contributes to aging, actually, right? Because like by the time you get to older age, your cells have been dividing for quite some time, and the pool of senescent cells, old cells that are not allowed to divide anymore, it gets more and more and more, right? And that contributes to the overall aging situations, if you would. Yes, Michael, go ahead, please.
Uh, so when the end of the primers, like when it falls off, right?
Mhm.
Does the polymer fall off with it, or does it get cut off? Like, does it like grow back, or is it just getting like shorter and shorter each time it falls?
Yeah, so it's not like it falls off, but look, right? If we look at this picture here, the okay, yeah, I get what you're saying, right? So the primer it does fall off the primer because it's RNA and it's not glued together by ligase. So eventually, when this is all said and done, you basically will lose this part right here. Okay, so that's gone. So remember, the black is the original, and then the blue is the copy. So you see the copy actually gets shorter, right? But that's okay because this part here, that got lost, that got fallen off or whatever. That part is just the telomeres. Okay, so let's say the important DNA starts like right here, right? Um, let's say this is actual coding DNA. Okay, so all these stuff prior, that's just part of like the telomere. Okay. Does that make sense? All right. Okay. Let's talk about Dolly. Has anybody heard of Dolly before? It's like I mean it's almost 30 years ago. So I was like wondering like some of you are probably like younger than that. That's usually the demographics of the program. So for those of you who, it's quite a famous sheep. For those of you who have not heard of Dolly, she was the first mammal to be cloned from an adult somatic cell, and the process that they use is called nuclear transfer. I'll explain what all that means in a second. The sheep, the clone sheep, Dolly, only lived for 6 years, half of the expected life span of a sheep. And let's take a look at why that is the situation. How many sheep do you think it took to make Dolly? Okay? Not counting the fail ones where sheeps die. But the actual successful um, time, how many sheep does it actually take to clone a sheep? I guess that's my question. Anybody want to take a wild guess? Yeah, one, two. Yeah, anyone else? Just a random guess. Oh, one. Okay, yeah, two. Yeah, let's find out. Okay, so here we go. Cloning Dolly. It's called nuclear transfer. Right? So let's start with one sheep. Okay, draw your best sheep. So this is sheep number one, and it is egg donor. egg donor. Okay, so what they did was they took an egg from the sheep. One time, a student got really confused. She asked me, "I didn't know sheep lay eggs." And then I was like, "No, not like a chicken egg, but like, eggs like, humans have eggs as well." So this is like sperm and egg, okay? So took an egg from the sheep and removed the nucleus. And then so that's the egg. It's probably too big. That's an egg without the nucleus. So it's just like the eggshell, if you would. Okay. Normally, how much genetic material would you find in the egg of an organism? Like in the egg of a sheep, in the egg of a human, how much genetic material are you gonna expect? Let's put some numbers to it, okay? We have 46 chromosomes. How many chromosomes do you expect to see in a human egg? Okay. Humans have 46 chromosomes. How many chromosomes in egg and how many chromosomes in sperm, guys? Yes, Trisha, half is correct. 23 and 23, right? So when the sperm fertilizes the egg, you get the 46 back. Correct? Everybody familiar with this concept? Okay. So what they did was they took the egg and then they removed the nucleus. That nucleus only had half the genetic material. So, they threw it away. Then sheep number two. Sheep number two is the nucleus donor. So they took a somatic cell and extracted the nucleus. Okay. A somatic cell is any cell in the body except for the germ cells. There are two kinds of cells in the body, okay? Just like a side note here, germ cell and somatic cell. Germ cells are cells that will become egg or sperm. Okay? You'll find them in the ovaries or testicles. Somatic cells are every other cell in the body. Liver cell, muscle cell, brain cells, heart cells, kidney cells. All these other cells that are not going to become, not going to become sperm and egg, they're going to be somatic cells. Okay. So they took a somatic cell, which contains the full set of chromosome from the nuclear donor, and then they extracted the nucleus. So there's the nucleus. Nucleus with full chromosome set. Okay. Sheep, I don't remember how many chromosomes they have, but it's not 46. But doesn't matter. It's the full set. Okay. And what they did next was they combined the two. They took the nucleus from the nuclear donor and then they put it into the empty eggshell. So this process simulate the fertilization. Okay, normally the egg has half. You gotta wait for the sperm that brings the other half, and then it creates the full set. However, in this case, because the nucleus already have the full set, and when you put it into the empty egg, it's like as if it got fertilized. Okay, but no male was involved in this process. Cheyenne has a question. So does that mean the eggshell yeah, because the nucleus was removed, right? So it had no chromosome at all, and then you are supplying the full set from the nuclear donor. Okay? So for those of you who wonder how they inject the nucleus into the egg, they actually just do it under the microscope. You might have seen it before in like TV shows and stuff like that. Like, they have this super big pipette that it looks super big under the microscope, and then it's like with an egg here, and then they actually have a needle that goes in. You might have seen some scene like that, right? It's what they do in IVFs, right? In vitro fertilization. Okay, so that's that. Right? They put it in. So that's two sheeps. Next, we need to bring in a third one. Anybody want to guess what the third sheep is here for? What's the role of this third sheep? Anybody want to guess? No guesses? Well, it's not Dolly yet. Okay, we're gonna make Dolly. That's right, it's gonna carry the egg. So the egg is gonna be put into this third sheep. What do we call people who carry babies for other people? Like if a couple cannot have babies, they can hire someone to do it for them, right? A surrogate, that's right. So this is a surrogate, which carries the baby, the fertilized egg, to be precise. And then you get Dolly. Okay, Dolly right here. All right, so it took 3 sheep to clone a sheep using this method. I have a question for you. 3 sheep was involved. Dolly is a clone of which one? Is it A, B, or C? Is Dolly genetically identical to egg donor, nuclear donor, or surrogate mother? Okay, well, let me share the results. The correct answer is the nucleus donor, right? Because that's the only sheep out of the 3 that is providing the genetic material, right? A didn't provide any genetic material. It provided the eggshell, but the nucleus was removed, so there is no DNA in it. Surrogate provides the womb, also no DNA. So Dolly is a clone of B. Okay, the nucleus donor. All right, so what does any of this have to do with what we are learning? Well, Dolly only lived 6 years as opposed to, the 12, 13 years of a normal sheep life expectancy, right? So how come? Well, the nucleus donor, which is the sheep that they took the nucleus from, was an adult sheep. Okay? So this sheep, if you look at its cells had been dividing for some time. Right? Because it's an adult sheep, right? It's gone through, I don't know, like 5, 6 years of life already. So, its cells has been dividing for some time. And so the telomeres in those cells are going to be shorter. Okay. Remember, every time you divide, the telomeres get shorter and shorter and shorter, right? So when they extracted the nucleus from the donor and use it to grow Dolly, when Dolly was born, even though it's a baby sheep, so like age-wise, it is age 0, like a newborn, but genetically, the age would be same as sheep B because it inherit the same telomere length as sheep B. Right? So because those telomeres are already short, is not going to be able to divide many more times, right? And so on a cellular level, on a genetic level, it is an old sheep. And that's why it's only going to be able to live, 6 years as opposed to full life. Does that make sense? Yeah, that's a good point, Michael. Like, he's asking, hey, why not take a cell from a baby sheep, right? I don't think that was the goal. The goal was not to create like a sheep that's gonna live long life. That was not the goal of the experiment. They were just trying to like clone a sheep in general. So, but yes, theoretically that would make Dolly live longer if had they taken the cell from like a younger sheep. So are we together on this concept? Right. You should have a question for me though about making babies at this point. After hearing all this, you should have a question about reproduction. No? Sheep was already 6 years old if their full lifespan is 12. Yeah, no. So what I meant, oh, sheep B was already six if their full life. I don't know what the age of sheep B was. Okay, like maybe it was, I don't know. Let's just say seven already. Okay, so then Dolly, when it was born, genetically, it's already age seven, so it lives 6 more years, which would be like 13 or something, right? Does that make sense? Like or like if it was already six, then 6 plus 6 gives you like 12 or something, right? Okay, the point is it's genetically old. Yeah. What are you guys laughing at? I was wondering if we can do it with humans. Oh, yeah. Well, I mean, ethically, they're not gonna approve cloning of humans, right? But they are working on cloning organs, though. Okay, because there's a shortage of like transplant organs, right? So, if you clone organs using your own cells, that would totally solve that problem. Anyway, I'm gonna point out the problem here. Okay, so. I don't know what age you guys are at. I'm guessing somewhere between 20 to, 35 maybe. Okay. So like your cells has been dividing for the last 20 years, okay? And your telomeres are going to be short compared to when you were born, right? So when you decide to have a kid, right? What's preventing you from passing on your short telomeres? Right? Like, wouldn't you pass on short ones and then your kid grows up and then they pass on short ones and each generation it would get even shorter and shorter and eventually like are we not going to be able to have kids anymore? Do you understand the problem? So obviously that's not a problem because humans population, if anything, it's like booming, even though the birth rate in Canada has been dropping. But globally, we have like 8 billion people now, right? So obviously this is not a problem. And why is it not a problem? It has to do with an enzyme called telomerase. Okay, so do not confuse telomerase with telomeres. Telomeres are non-coding repeats, the TTAGGG, that's located at the ends. And then the telomerase is an enzyme that is being expressed in germ cells. Okay. So an enzyme, of course, is proteins, right? So your cell, the germ cells are going to be producing this thing called telomerase. Without telomerase, the telomeres, which are represented by these red caps here, it gets shorter every time the cell divides. But with telomerase, which is what these triangle things are, they are able to prevent the telomeres from getting shorter. Okay, they re-lengthen the telomeres. Right? So when you mix sperm or eggs, which is what germ cells are, okay, germ cells will become sperm in male and eggs in female. When you do that, the telomerase will reset the telomeres. So when you pass it on to the next generation, your children are going to have brand new telomeres. And then as they grow, as their cell divides, then it will shorten in their body. Okay. But every time you have a kid, every time we restart a new generation, you reset the length of the telomeres because of the telomerase. Now, why did that not happen in the case of Dolly? Well, because Dolly was not conceived with sperm and egg, right? It was the cloned using an adult cell. That cell does not have telomerase in it because it's not a germ cell, and therefore it did not re-lengthen the telomeres. And so it inherited the genetic age of the adult sheep. Do you understand? Any questions at all? Because this is about to go even more crazy. So please ask away if you have anything. Okay. Well, I hope you are enjoying the topic so far. I think it's quite interesting stuff to know. Now, if you are with me up to this point, there's a question. Human surrogacy isn't same as cloning. Absolutely not. Okay. Surrogacy is just, if a woman has like, for example, like a damaged reproductive system, right? Like maybe the uterus is not able to carry the baby, but she still have eggs, right? So they can still take the eggs and the sperm from the father and fertilize it outside of the body, and then implant it into a third person, a woman, who is has agreed to carry the baby for them. Okay, but that surrogate mother does not contribute any genetic information. Yeah, I think I read something. You guys can Google this. There was a mother whose daughter cannot carry a baby, so the mother agreed to be her surrogate. So which means the grandmother is carrying the grandchildren for the daughter, right? Like, do you guys follow what I'm saying, right? Like, so that is quite a an amazing gesture, right? So you can Google that if you're interested. Okay, so back to my story. What was I saying? Sorry, lost my train of thought. Yes, if you are with me up to this point of the lecture, then you understand that every time we copy the DNA, it gets shorter. If they become critically short, then the cell cannot divide anymore, and that contributes to aging, basically, right? Now, telomerase is able to fix that when we have kids, right? It resets the telomere length and then it pass on brand new ones to the next generation. So it's logical to think that telomerase might have anti-aging property. In nature, telomerase is only expressed in germ cells and stem cells as well, but let's focus on the germ cells. Um, then what if we turn on telomerase in regular cells? Okay, what if we turn it on in like our bodily cells? Is that going to delay the aging process? Is that going to allow the cell to divide more times and maybe even reverse some of the aging effects? Well, it turns out that's not going to happen because what we found was that when telomerase is turned on in regular cells, in somatic cells, it actually caused the cells to turn cancerous. Okay. Now, I want to backtrack a little bit and explain what I mean by turn on and turn off. All right. You have many cells in the body, trillions of cells. All of the cells everywhere in the body carries the exact same DNA. Like your brain cell is going to have the same set of DNA as your kidney cells, as your muscle cell, so on and so forth. So all those DNA are instructions to make a bunch of proteins, right? Okay, so think of the DNA as like a recipe book with hundreds of recipes in it. And every single cell has that same book. But here is the difference between all the cells. The brain cells are going to need different proteins than the liver cell, right? And so the proteins that you'll be making in the brain cells, the recipes that you'll be using in the brain cells are going to be different from the ones that you'll be using in liver. So because telomerase is a protein, we need a recipe to make it. We need a DNA to make it. So all the cells have the recipe to make the telomerase, but whether you make it or not, whether you turn it on or off, is going to be dependent on what cell you are talking about. So it turns out for telomerase, you only turn it on, you're only going to cook the recipe in the germ cells. in your bodily cells, you're not going to turn it on, even though you have the recipe for it. Okay? But as we will learn later on in the lecture, I think, or next week, right? Sometimes there are changes that happens within the cell, and you turn on the gene in a cell that it's not supposed to be turned on. Okay? You are making the recipe in a cell that's not supposed to be making it. Okay? And so in this case, we have a cell, regular cell, that's making telomerase when it's not supposed to. So what's the consequences? Well, that cell will not have the shortening of the telomeres anymore. When it gets critically short, it's supposed to stop dividing. It's supposed to be a signal that causes the cell to go into growth arrest. But because of telomerase that get turned on accidentally, that cell can now keep on dividing and dividing and dividing with no control. And that creates a tumor. Uncontrolled cell divisions is basically a collection of cells now. And that tumor could be cancerous. Okay? So researchers are trying to explore telomerase inhibition as a potential treatment for cancer. If we can block the telomerase in these uncontrolled cells, then maybe we could stop its growth, right? Maybe we could shrink the tumor, and it could be a useful treatment for cancer. Any other questions? Oh, there's a question actually I didn't see. Telomerase does nothing for us besides passing it on to our kids. Yeah, pretty much, right? Like it lengthens the telomeres so that we can, ensure uh, it gets passed on in a nice way for the next generation. So tumors are due to telomerase turning in the wrong cell. Exactly. That's one of the causes. Okay. We will learn that there are many different causes. We will learn 6 causes of cancer. This is one of the 6. Another question. So by anti-aging, yeah, exactly, right? That's the thing, right? Why do we age? Actually, aging might be a mechanism to counter diseases like cancer. Okay, so as people live longer, the chance of getting cancer is actually higher, right? So cancer, of course, I understand cancer can happen in kids as well, but for the most part, cancer is a disease of the aging population, right? So as we live longer and longer, as people live over 100 years old, right, the chance of getting cancer is going to be higher. So it's like a trade-off, right, between longevity and disease, right? Are we together? Thank you. All right, just a couple more slides and then we're gonna stop. Okay, there are some poll questions here. I'm just gonna give you the answer because I don't have time to go through it with you at the moment. The answer is B. Just write down. Okay, B again. BBD, and then A. BBDA. Right, just write down somewhere, and then you can do it later when you've forgotten the answers, and then you can check it again. Okay, BBDA. Okay. Next, we're going to talk about the cell cycle and mitosis. Probably don't have time to go into details about it, but I'll give you an introduction to it. In lecture 3, we learned about the cell theory, right? And one part of the cell theory state that all living cells come from division of pre-existing cell. Nobody really know where the first cell came from. Right? People have theories about it, but no one knows for sure because no one was there when it happened. But we know to get new cells, you need to divide an existing cell. Okay, and so why do cells divide? Well, one reason is to replace lost or damaged cells. If you have a cut in the finger, you've damaged some cells, you've lost some cells, and cell division is going to basically bring them back. Okay. Growth and development. You have a lot more cells now than when you were a baby. All those extra cell came from the division of pre-existing cell. Reproduction, okay? We are sexual, uh, we do sexual reproduction, okay? So we do not rely on cell division to reproduce. But some organisms, they are asexual. They are asexual. They reproduce asexually. And what that means is they do not require fertilization of egg by a sperm, and they just do the division to make more copies of themselves. Okay, so the cell division is actually called mitosis. Don't worry about that right now. We're gonna go into more details next time. But these are some organisms that can reproduce asexually. They don't need a partner, and it happens rather fast. A bacteria like E. coli, under ideal conditions, can make copies of themselves with just under 20 minutes. Okay, so that's really fast and it requires less energy. Think about humans, right? Like how long does it take to carry the baby to term and then raise the baby, right? It's like 10 months just to carry the baby, lots of energy, resources dedicated to it. So definitely asexual reproduction in that regard, it's much more advantageous. Did you know if you take a starfish, sea star, and you like cut it in half, for example, you're gonna get two in the end, okay? That's a type of asexual reproduction. It's not the same as earthworms, okay? Worm, if you cut the worm in half, not that you should, but if you did, the head is going to grow a tail, but the tail is not going to grow the head back, right? Okay, so that's not reproduction. That's just regeneration, right? But starfish is different. Starfish, each half is going to be able to, create a full starfish. Obviously, there's like a minimum amount of mass that you need. It's not like you chop off all the arms and you're gonna get like 6 starfish or something like that. It doesn't work like that. There has to be some minimal cell mass for this to work, right? But for the most part, it reproduces asexually. Plants can do that, right? I have a lot of like aloe vera plants at home, and then people always ask me for it, and I just break off a piece and they take it home, and then they can grow their own, right? And then there's amoeba and then something called a hydra. Don't worry about it, okay? But these are things that could reproduce asexually just by cell division, okay? We do not do reproduction this way, right? We do sexual reproduction. You need a partner. You need two individuals to contribute the genetic information to make the babies. The disadvantage of asexual reproduction is that it has low genetic diversity. Right? If there is some kind of mutation, some kind of changes in the DNA, that change is going to be preserved in all of the offsprings. Because every time you divide, you're copying exactly the same thing, right? So if there's a bad mutation, that same bad mutation will continue to be propagated through generations. Okay. So there are 2 stars here. Later on, I will tell you, rapid division is actually a way to introduce genetic diversity, but that's going to come later. Okay, just like keep that in mind for now, I suppose. I just want to refresh your memory on DNA structure. Right, we know DNA is the double helix, but we don't really find DNA on its own within our cell. Because it's not going to fit, right? You got like 2 meters of DNA and you try to fit it all into a tiny cell that's a couple micrometer wide. You got to package the DNA. And the way to package them is to wrap them around these proteins. Histone is just proteins here. And it creates the chromatin. Okay, and that's the state of DNA inside a nucleus. Okay, we wrote this down in lecture 3. When you try to divide a cell, then you need to move the DNA around, and in order to do so efficiently and without tangling them, you gotta package the chromatin even further, and that's when you get chromosome. Okay, so chromosome, chromatin, and DNA are basically all DNA, but it's just different levels of condensation. Okay, the most condensed form is the chromosome. the less condensed version is the chromatin. If it completely unravels, then that's just the naked DNA. But that, like I said, it doesn't really exist on its own, right? You will do that when you do DNA replication, or like transcription, but DNA is rarely gonna be completely by itself. So different organisms have different number of chromosomes, and humans, of course, have 23 pairs of chromosomes, 23 from each parent, right? 23 from mom, 23 from dad, and that makes up a total of 46 chromosomes. Of the 23 pairs of chromosomes, 22 pairs of them are autosomal chromosomes. Autosomal chromosomes, the short form is autosomes. These are chromosomes that determine various features of you. Right? So things like skin color, eye color, blood type, the kind of hairline you have. You know, some people have straight hairline, some people have this, it's called like a wiggle peak, right? Your ears might be attached. Your ears might be have a lobe. I have a lobe. Some people have attached, right? That's genetically determined. All these features about you are going to be encoded within the autosomes, okay? 22 of them, okay? So these are the chromosomes that contains the instruction to make all these proteins, and then these proteins are going to determine, some of these features of yours, okay? The last pair, 23, 23rd, the 23rd pair is the sex chromosome, and as the name implies, it determines the gender of the baby, the sex of the baby, right? If it's XX, then it's going to be a female baby. If it's going to be XY, then it's going to be a man, a boy baby, okay? Grow up to be a man. So who determines the gender of the baby? Is it the father or is it the mother? What do you guys think, huh? Yeah, it's the father, right? Because the father is XY, so half the sperm will carry the X, the other half of the sperm will carry the Y, right? An egg will always be carrying the X. So if an X sperm fertilizes the egg, then it's going to be a girl. If the Y fertilizes the egg, then it's going to be a boy. And the chance is 50-50. It's like flipping a coin, right? It's just a quick funny story. My grandmother, she's 90 something, very traditional, right? And every now and then, when we have gatherings, she would sit beside me and whisper into my ears. I have two daughters, right? You probably know that already. And then she would whisper into my ears, like, why is your wife not giving you any sons? And then I would try to explain to her every time that actually it's not her fault, right? It's actually my fault. It's the guy that determines whether the baby is boy or girl. And then she'd be like, no, 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 that's not true. Like I would know because I have many sons, right? And so every time I try to have this conversation where I try to convince her. And then she would be like, and besides, you have two girls already. If you have another kid, I know the next one is going to be a boy, right? But as you know, it's not true, right? The events are independent. If you flip a coin like 5 times and you get 5 heads, what's the probability that you're going to get a head again? It's still 50%, right? Each event is independent of what happened before, right? So even though you have a lot of daughters, there's no guarantee the next one is going to be a boy either. Okay, so there is the autosomes and the sex chromosome. Last slide. Chromosome number in animals and plants, there is no correlation, it seems, between the size of the organism and how many chromosomes they have. Something as small as a goldfish can have 100 chromosome, whereas something as big as an elephant, it's 56 chromosome. Okay, so there's no correlation uh, at all. And I think that would be a good place to stop for today. We will try. I think we will definitely finish. We have like 37 more slides to go. Sounds like a lot, but not really. We'll be able to finish next time, and we probably will also start um, lecture 7 on the Tuesday. So please print those sheets as well. Can animals and plants have trisomy 21? Trisomy 21 is a Down syndrome, which is a condition for humans. Animals and plants can have trisomy. Trisomy just means like an extra chromosome. They can have extra chromosomes, but it's not going to necessarily lead to Down syndrome. It might be, some other conditions that they might have. All right. That's it. Thank you, guys. Enjoy the rest of the week. Don't forget to change your clock this Sunday. We are turning back for the daylight saving. Have a good one. Bye bye.